fat-free, low-fat, low-sugar, low-calorie, zero-calorie, zero-sugar, keto, paleo, vegan. These are all steps that 45% of the global population take while trying to lose weight, according to Ipsos. And according to the World Health Organization, 1 in 8 people are obese. What if I told you that hydrangeas can help prevent obesity? Wait, Eva, you mean the flowers that change color depending on the pH of their soil, the kind we learned about in AP chemistry? Yes, the sepals of the hydrangea flowering plant shift to a rose pink when the soil is alkaline, and then to a fiery blue when the soil is more acidic. In fact, according to a study by Yoshida et al., this process is controlled by the same genes that are involved in anthocyanin biosynthesis, or more simply, the production of anthocyanin. Anthocyanin is essentially a flavonoid or phytochemical compound that exhibits beneficial health properties as an antioxidant, immune response regulant, anti-inflammatory aid, membrane strengthener, and numerous others. In addition to their use in studying successful outcomes in ancient and modern folk medicine, anthocyanins can also lead to the prevention of obesity and diabetes cases, as observed by mice by Tsuda et al. So how can some flowers reduce the instances of obesity in the world? By genetic modification. Don't worry, CRISPR is a contemporary gene editing tool used by scientists to learn about gene expression. Basically, formed from a bacterial immune response, CRISPR, or clustered regularly interspaced short palindromic repeats, releases an enzyme called Cas9 to clip a targeted segment of DNA specified in CRISPR's RNA so that reproduction is manipulated. This also works with the addition of a DNA sequence as well. Essentially, this procedure dictates that a primer be made for the construction of the guide RNA for CRISPR using previously researched data on specific genes associated with hydrangea colored shifts. Then CRISPR will remove those sequences or add inhibitors for anthocyanin biosynthesis in hydrangeas in group 1 and add more sequences or promoters in group 2 while leaving group 3 as control. Then for observation, the hydrangeas will be planted and sepal color will be observed depending on the changes in soil pH to provide a more accurate indication of whether anthocyanin was produced. If successful, group 2 will present stronger hues of blue and red in hydrangea sepals, whilst group 1 will be paler. The next step in this research process would be to apply the genetically increased production of anthocyanin to foods, such as raspberries, and measure the content in order to reduce instances of health issues related to nutrition by increasing the production of anthocyanin.